microscope. Okay, first of all, we'll talk about what, guys? A light microscope, and in that, first of all, let's see the structure. In the structure, what we have, a base on which that particular one is there. Okay, a base is there in this particular one which holds the various parts of the microscope, such as the light source, the down one, the light source, the fine and the coarse adjustment screws or knobs, they are also on the base. Okay, so this particular thing is termed as what? Base. After that, you have a C-shaped arm. You have a C-shaped arm by which you hold it. Okay, and this particular one basically connects the ocular lens to the objective lens. What it does do? It connects the ocular lens to the objective lens. That is the main function of the C-shaped arm, and it helps to hold the microscope. Then comes the mechanical stage. Okay, in the mechanical stage, what you have, an arm is there. An arm is there which bears the stage with stage clips. Clips are there to hold the slide. Okay, to hold the slide, and basically because slide can move now here and there, to hold them you have these particular clips, and there is an aperture in this mechanical stage at the center which permits the light to reach the object from the bottom. So in this mechanical stage, what you have, guys, you have an aperture through which the light can come from down up, okay, to the objective lenses and later on to the ocular lenses. Is it fine? Then after that, if we talk about the magnifying stuffs, just now we have done that the ocular lens is there, that is termed as the eyepiece, which is having the power of 10x, and then you have those objective ones, that is 4x, 10x, okay, 100x, and that particular 40x also, that is a high one. Fine. So that is termed as ocular and objective lenses. Ocular lenses through which you observe, and objective lenses are the one through which the light goes towards the ocular lenses. So that is termed as ocular lenses magnifying stuff. Then, as far as the illuminating part is concerned, in the illuminating part we have condenser. And what is the use of condenser? It is beneath the stage, down in the stage, which focuses a cone of light on the slide. What it does? C for C, you remember. That it focuses what? Cone of light on the slide. After that comes what? Iris and diaphragm. Important. It has been asked in NEET PG also. It controls the light that passes through the condenser. So who is the controller of the light which passes through the condenser? That is the iris and diaphragm. Okay. So condenser is there beneath the stage, which produces what? Cone of light on that particular slide. Fine. So basically C for C, you remember. And which particular thing is there which controls the light passing through this condenser? That is the iris and diaphragm. Is it fine? The light source might be the natural source like sunlight and all, and you have a mirror that will reflect it. So that is there. Or nowadays, what is coming in the microscopes? The electric bulbs. Okay, so the LEDs and all, they are coming up. That will be sent like that. Fine. Along with that, another important thing is those fine and coarse adjustment knobs. Okay, and what is the role of them? They sharpen the image. What they do? They sharpen the image. So in total, what you have observed, what is there in the light microscope, guys? So in total, you have all those particular things, like for the magnifying stuff, you have these ocular lenses and these objective lenses of different powers. You have a stage, okay, over which it's you can hold it up. Then you have C-shaped arm, then mechanical stage having a aperture, and later on those particular things like condenser, okay, cone of light, C for C I told you, iris and diaphragm which controls the light which pass through condenser, iris and diaphragm, that is being asked also. Along with that, the light source can be a mirror of the natural light, or it can be nowadays the electric bulb and all. And besides that, you have the fine and the coarse adjustment knobs, which are there in this particular light microscope. And if it is having a single one, so uniocular, and if it is having two, binocular. Okay, so that is termed as a light microscope or a bright microscope. Is it fine? So this is commonly used up in the practicals also. So you should know the sound knowledge of this particular one. And it is being mostly done for gram staining, Zn staining, which you will be doing the practicals, and we will discussing that particular one.